Hi everyone, my name is Alona and I will present to you our work called Simultaneous Detection and Classification of Partially and Weakly Supervised Cells. This work is a collaboration between the CS and medical departments in the Technion with Rambam Medical Center. So before we dive into the details, here's a brief view of what we shall see in this talk. So we start from a biopsy extracted from the bone marrow sample of a diagnosed patient with multiple myeloma. The bone marrow smear is then transferred to glass slides, stained and scanned with a high-resolution scanner. The result is a whole slide image. The whole slide image can be broken into smaller sized patches called tiles, which serve as the input for our deep neural network. The results of our training scheme, which I will next describe, is a classification of all cells in the image. Our final goal is outputting classification results for each and every tile and aggregating the cells in a histogram. The histogram acts as a footprint for each patient, describing their susceptibility to a given treatment. So our work is a small step towards personalized medicine, where a patient undergoes different tests and analyses, after which they are offered the best treatment that will provide a positive outcome. So in this work, we will concentrate on multiple myeloma, a disease of the immune plasma cells, which accounts for 2 to 3% of all human cancers. While multiple myeloma patients now enjoy from longer remission periods thanks to proteasome inhibitors, the disease often returns with higher resistance to treatment. So there have been studies that have shown that even a small percentage of harder to treat cells can lead to a relapse of the disease. In order to detect such cases in advance, a cell-level analysis of pathology image is critical. While analysis in the slide or tile level are the bread and butter of modern pathology, analysis at the cell level is impractical for humans. Our aim is thus automatically analyzing a bone marrow sample and constructing a patient-specific profile that will aid diagnosis and treatment planning. So deep neural networks have become the gold standard for applications in computational pathology. Learning-based approaches operate on 3 abstract levels. The first group operates on the slide level, annotating each slide with a label. For example, in breast cancer, there exist 3 biomarkers: estrogen, progesterone, and HER2. A DNN can predict the result, either positive or negative, for each biomarker, very similarly to a human pathologist. Next, one can operate at the tile level. Recall, a whole slide image consists of smaller sized images called tiles. In this way, one can infer the cancerous regions within a slide while putting slightly more effort into the annotation process. While this method provides more detail, it disregards the intra-tile cellular connections. Finally, one can operate in the cell level, i.e., localizing and characterizing individual cells in pathology images. While this is the ultimate level of detail, it requires precise and expensive human annotations. Most approaches in this sub-area are concentrated on the accurate segmentation of cells, disregarding any downstream tasks. So this leads us to our approach. Given a slide of a bone marrow smear, we would like to construct a patient-specific histogram containing the accurate cell composition of the patient. This can alert the pathologist on the existence of violent types of cells, which directly impact the treatment type and expected outcome. This histogram representation requires analysis of each tile at the cell level, and thus can be done once the cells are detected and correctly classified. So to alleviate the bottleneck of individual cell annotation, we define that each training image includes an average of 5% of annotated cells. The annotations are either points or bounding boxes from which the center pixel is extracted with corresponding classification label. We divide the overall cells in multiple myeloma immunohistochemistry image to 9 classes, which represent different amounts of the proteasome protein infiltrating the cells. 4 stands for most healthy cells, in which the proteasome is located in the cell nucleus, and -4 stands for the most violent cells, where the proteasome has expanded to the cell cytosol. Finally, to perform evaluation, mainly of the detection capabilities of our approach, we add an additional set of images in which all cell centers are annotated, but without the corresponding class. So once a human annotator has annotated 5% of the cells in each image, we can compute the label encoding that will be more helpful in training the DNN. For each image, we construct a cube of the same spatial dimensions times the number of classes. In each class channel, we create a ball of radius R1 around each point from that class. Around each ball, regardless of the class, a ring of radii R1 and R2 is added in the background channel. This enforces the cells to be separate from each other and reduces the chance of cell clustering. The final encoding is a cube of the same spatial dimensions as the input image with depth equal to the number of classes. The network architecture is a unit with a ResNet encoder pre-trained on ImageNet. The network receives the input image and corresponding label encoding and is trained to output C probability maps, where C is the number of classes. As opposed to semantic segmentation, where we have the fully supervised labels during training, here we have a weakly and semi-supervised label encoding. The shortage in annotations is alleviated via the specialized loss functions. Our loss function consists of two terms. The first, partial cross-entropy, takes into account the network prediction only in the annotated regions omega L. The label encoding serves as one hot matrix M, and the network output probabilities are denoted as Y. An additional class-dependent scalar gamma is added to increase the effect of the cell classes versus the background. As the partial cross-entropy loss takes account only a small portion of the pixels, we add a regularization term, which enforces smoothness of the output probabilities. Here, Yi and Yj denote output probability values for adjacent pixels, and Wij is a weight function producing larger weights for similar colored pixels. Once the network has finished training, inference over a new image is performed as follows. First, we sum the output probability images for the cell classes without the background, then perform thresholding and remove smaller and larger sized cells. Finally, for each obtained blob, we compute the average probability over its coordinates. The class that achieves the most maximum result is chosen. This repeats for all detected blobs. So let's take a look at the numeric results. The second line corresponds to our method with the label encoding I have previously described. The first line corresponds to the baseline. The baseline entails performing first detection of the cells in the image, then accurate segmentation, and finally, given a bounding box of the resulting blob, performing classification with a small CNN. One can see that the combined performance, F combined, of both cell detection and classification in the rightmost column. Our method improves upon the baseline, this because it performs both tasks with a single network, reducing cumulative errors which can occur from one network to the next. Another issue we have discovered is that of intra-annotator confusion. One can see a confusion matrix on the right showing the performance of our method. The most common mistakes of the model are between adjacent classes. This can be expected since class 2, for example, is very similar to class 3. Thus, the original annotation data can be noisy. On the left, we can see the Fscore if we disregard misclassifications from ±1 adjacent classes. One can see a better Fscore reaching almost 85%. Let's take a look at the qualitative results. On the left, one can see the performance of the baseline model versus ours on the right. On the right, we display the classification performance. Each cell is colored by its predicted class, where you can see the meaning of the classes on the left. Recall, -4 are the most violent myeloma cells, and the Grouchoth boxes are shown as bounding boxes. Finally, we started from our final goal of creating a patient-specific cell profile to be used for diagnostic purposes and treatment planning. Here we see 3 slides for patients with an increasing gravity of multiple myeloma. In the first, the cells are mostly nuclear, as can be seen in the histogram, whereas in the bottom, there is a large amount of cytosolic cells, implying the disease may be harder to treat. So we hope this method will allow faster development of datasets for additional cancer types and overall push the frontier of personalized medicine. Thank you for listening. Any questions?